So today, lab 4, but before we really get into lab 4, look at your streak plate from last week, right? One of your first things was to draw and record your results from that. I think just about everybody has isolated colonies, right? I came around and looked at them as I was handing them out. Anyone not have isolated colonies? I think of maybe just a few of you, where the colonies are not separated from each other, right? Your little circles are touching, right? It was somebody. Daniel, was it you? No, you got isolation. Who is it that I said didn't really have isolation? Yeah. Okay, yeah. So, see how close, we'll pass hers around. I'm going to actually tape it shut so it doesn't get popped open. I highly suggest looking at Megan's plate. You'll probably see it again. Hint, hint. See how all her colonies are really close to each other. She doesn't really have any isolation. That's not necessarily her fault, except for the fact that why does she have some growth in the other ones? What could have gone wrong? And I'm away from my microphone. Why wouldn't she have gotten isolated colonies? Yeah, so she may not have picked up any from the previous section. Her loop might have been too hot and she might have killed what she picked up, so she was just spreading dead cells so they didn't grow, right? She may have forgot to do the step. I don't think that's the case. I can kind of see streak marks on her plate. She get an F for today? No, she gets an A for effort, right? Because remember, we're learning. And even sometimes, remember the beginning plates that I gave you guys with the Klebsiella pneumoniae? It was supposed to be a streak plate, and we just had a little bit of growth on those plates. The organism just didn't grow well. You know, that wasn't any fault of the scientist who inoculated that plate. And sometimes you're just going to have too much bacteria, and you just don't have enough space to spread them all out, right? So this is a technique that even if you've done it 20 times, you still sometimes aren't going to come out with a perfect streak plate. Picture perfect. Does that make sense? Okay, so look around and see how this one doesn't have what we want, but that's okay. You would just do it again. So after about 24 hours of growth at 37 degrees Celsius, how many cells would be in that colony? So that one little isolated colony, about how many cells do you think are there in your little isolated colonies? Millions? Could even be billions, right? So for a particular species, you would have to know. And so for instance, for E. coli, under ideal conditions in like a broth culture where they're being shaken up, right, and they're getting aeration and getting away from their yucky waste and they're getting all the new fresh food, it can double itself every 20 minutes. So let's just round that up to a half hour, right, because that's nice, easy math, right? And we're dealing with a plate, so that's not ideal conditions, right? They're not swimming around in a liquid broth. You okay, Megan? I wasn't picking on you or anything, okay? All right, so per hour, right, so every 24 hours, every half hour, right, after 24 hours, we'd have 48 doublings, right? So they have doubled 48 times. So you went from 2 cells to 4 to 8 to 16 to 32, right? So we could mathematically calculate this, right? So that would be 2 to the 48th power. 2 times 2 times 2 times 2, right? It gets really big really fast. There are different ways you can mathematically play around with this without plugging it into your calculator, right? If you remember how exponents work, right? So we could essentially round it up to 50, right? And divide them up between 50 and 10. So then we just deal with this one section right there. So 2 to the 10th power is about 1,000, right? 1,000 bacteria. 2 to just the 10th power. Now, if we multiply that by 5, right? So we just substitute, right? We solve this, we just substitute that in there, right? So now 3 times 5 is 15. So if we separate this out into chunks of zeros that we recognize, right? 15, remember you can add exponents. So if we add 6 and 9, that's 15, right? So I can split them up like this. 10 to the 6th, 6 zeros, is what we commonly refer to as a million, right? To the 9th, that's a billion. So essentially around 100 billion cells could potentially be in that one little colony. Let that sink in for a second. That's a lot of bacteria, huh? Now I see why I say you just need a little bit, right? When you're taking your sample, you just need a little bit. And for some of you guys, that a little bit is hard with the loop, right? I'm going to suggest that you use a needle. So notice a lot less surface area, right? Where you can literally just pick up a tip. But imagine, right, how many can fit on that little tip of a needle? A lot of bacteria. More than enough for what we need to do. So if you were to actually calculate it out, it's 2.8 times 10 to the 14th power. Right? This calculation's off by about 3.5. A factor of 3.5. So, could we figure out how many are actually in a colony? Yeah. Yeah, you can. But I don't know about you guys, but would you want to actually count? Yeah, she's like, I'm not counting that high, so no, it can't be done. But, math isn't all about counting every individual cell, right? We can do calculations like we just did. You guys are going to do calculations later, right? Based on smaller sample sizes, right? Where you're diluting it out so that you're counting a smaller size, but again, that's a factor of how much you diluted it, right? So we don't, when they're testing the water supply, right? They don't test all the water in our municipal water system, do they? They take samples, right? And based on how many bacteria they have in those samples, gives us a general idea of how many are present in that larger water supply. Make sense? So we'll do, and again, it's going to depend on other factors. What organism are you working with? E. coli can grow really fast. Other organisms not so fast. What type of food did you give it? Some of them like certain types of food. Others won't even grow if you give them the wrong food, right? There's one that I give as an unknown, and I'm like, every semester, I'm like, y'all, you really need to pay attention to what food you're feeding it, because this particular one will only grow on TSA. If you accidentally put it on nutrient agar, you'll get no growth, because the nutrients it needs are not there, right? It needs the TSA. So that comes into play, right? The conditions, what's the temperature like, right? We definitely employ different temperatures in this lab, right? Is there oxygen present or not? And in what concentrations? So there's a lot of variables to take into consideration. And some bacteria species, as we know, can grow anaerobically, right? Without oxygen. So they can actually grow into the agar, right? Just as we noticed with the fungi, remember those filaments, the hyphae can grow into the agar. This is true for your bread and your cheese, right? You can't cut that stuff off, y'all. It's growing into it, okay? There's no cutting the block of cheese to remove the mold, right? You may be removing the stuff on the top, but not the stuff on the inside, not the microscopic stuff that's growing into the cheese itself, okay? So facultative anaerobes, these are ones that can grow aerobically and anaerobically, and they actually grow better with oxygen. So again, they're going to be affected on where they're growing and on that surface or in the media based on their different abilities. And they grow really fast aerobically, and so they'll generate lots of energy, lots of ATP. So the cells in the middle of the colony, think about this. They're not just growing out, they even grow up. You know, so they'll start stacking on top of each other too. And imagine those crowded conditions too. You're not going to get as much air as if you're at the top of the pile. All right, so that's going to affect their growth, right? It's not uniform throughout that colony. So, therefore, the doubling times are going to be different, especially when you're on an agar plate, as opposed to if they're in the liquid media and they're being shaken, where aeration is being complete throughout, right? Air is getting all throughout that tube. And you don't have that problem of oxygen or even nutrient availability because you're never at the top or the bottom of the pile, right? You're swimming all through the liquid. So, you could get, and there's been a study that's been done, this is a link to it, using E. coli, a particular strain of E. coli, and they found only about 3.3 times 10 to the 9th cells grew over that 24-hour period, although they didn't specify all their conditions in their paper. But again, to give you an idea, I mean, literally billions of bacteria are in one of those little circles on your plates, potentially. Right? It's a lot of bacteria. So, so that number suggests that it's taking them under those conditions on a plate about 45 minutes to double, as opposed to their maximum ability in a broth culture of about 20 minutes. So, can we determine this for ourselves? Yeah. We usually, in the past, have actually done the replica similar to this experiment. We're not going to do that this semester. But there are different ways that you could do it. You could pick up from the plate a colony, right, and dilute it out. The problem with some of the methods that we use is if you don't microscopically count them, if you use what a lot of scientists do, which is the plating and then counting. So you're gonna spread them out on a plate, and then you're gonna count how many colonies arise. When you do that, only the living cells will actually grow, right? Any dead cells you won't be counting, right? So that's just what we call a viable cell count. You'd have to actually use a special chamber called the hemocytometer, and we don't have any here yet, but hopefully the biotech program, I checked with them this weekend. As I was prepping the other day, I was like, ooh, you know, it'd be nice if I had one of those to at least show my students. But I have pictures in another lecture when we actually go into counting more. But it's very expensive. I looked it up. It's like $300 for this slide, because it has a special chamber that has a specific volume in it, so that when you put your sample in, you know the volume of the sample. And then it has a grid etched into the slide so that you can use it to help you count the cells, keep track of the cells that you're counting within that grid. So it's really kind of cool. It's called the hemocytometer for counting. And I've used them before for eukaryotic cells, which are much larger, right, and easier to count. But they can be used for bacteria as well. But as you can imagine, tedious process. She was like, oh yeah, I'm not signing up to count that. And depending on how many are in that sample, you know, you'd be counting a long time. So usually you dilute the sample, right? And you take into account that dilution to figure it out. So one way you could pick them up off of a plate too, right? But again, depending on the organisms, you might even have to gouge out the agar to make sure you get any that are growing into the agar, right? They don't just grow on the surface. Some of them can grow into the agar. Or you could use this method where you remove all the colonies off the plate, usually a plate containing about 100 colonies, and you take the average of that calculation. And so this is a cool video that shows how quickly E. coli can divide. Let's see if it'll go. Make it big. So they've sped this up, but look at these guys go. Before you know it, it's full.
For the hemocytometer,
Mm-hmm.
all cells are dead?
No, when you do a hemocytometer, you're counting all cells, living and dead. Unless you use a special stain, they have special stains available too that'll help you differentiate from the ones that were alive before you stain them, opposed to the ones that were dead. So you can ask that question using special stains. But if you're just counting whatever you see, you're counting living and dead, you don't know which is which. But if you grow them, then of course it's only the living, the live ones that will grow, and none of the dead ones will. So your count will be off if you wanted to know all of the organisms that were present in the beginning sample. So again, I'm not going to go over terminology, but we have like a little glossary for you to help you some of the terms that we're going to use as I go throughout the lecture today. So there's some terminology here that Peter Hickman, another one of the instructors that teaches this course sometimes, and I agree that we're not going to get into the real particulars of chromophore versus oxychrome, right? It's getting a little too insane about what part is doing what of the dyes that we use, right? So we're not going to get real particular. But we do have some pictures of the shapes of these dyes, just to give you an idea of what they would look like. Not by any means do we expect you to memorize them, but just give you an idea too of they typically will have these ring structures. That's what allows them to resonate, actually absorb and reflect color. That's why they have a color to them, these types of molecules. So there's a very subtle difference between crystal violet, which appears violet purplish color to us, and malachite green. It's just this one little dimethylamine group, right? The rest of the molecules are exactly the same. It's kind of crazy. So resonance and stuff, right? This is chemistry type stuff. Some people, I say chemistry and you go, right? But chemistry is how the world works, y'all. It's really how it works. So again, it's the interactions of these molecules is why we see the different colors that we do. So for those of you guys that love the chemistry, right, it's there for you, but you don't need to know it for this course. So the important thing here, though, for these guys, I'm going to see where I have it highlighted. So again, malachite green is because it's where it's absorbing the light at what frequency it appears green. And then of course, crystal violet's on this other side, and so therefore it appears violet in color. Both of these dyes, though, notice are positively charged. You see the positive here, and then they usually have negative ions associated with them. Usually it's a chloride ion. So these, in solid form, they combine with the chloride ions and they form these crystals. Not all that different like when you think of salt, right? Sodium and chloride. But then when you put them into solution, when you put them into a liquid, they disassociate. So this becomes positively charged, right? And this chlorine ion is negatively charged on its own in the solution. So it's this positive charge that's really beneficial to us in the solution. The dyes that we use are in solution because that is going to interact with the cells. That positive charge is going to attract it to our cells. And our cells are negatively charged. So opposites attract. Most of the dyes we use are positively charged, what are called basic dyes, right? So basic refers to acid and base kind of things, right? And in this case, it attracts hydrogen ions. It's positively charged. So gram stain we're going to use today in a very simple stain just to stain cells, but we'll also use it in conjunction with other stains and a multi-step procedure when we do the gram stain next week, right? And then malachite green, which is another basic dye, we'll use it when we stain endospores the following week. And again, though, both basic positively charged dyes, they're going to be attracted to these cells that are negative, or the cellular components that are negative. Right. So why do we use our microscopes? Our microscopes are going to help us improve resolution, right? Something you guys need to be able to calculate. Remember that formula, right? So we take into account, especially with visible light, how it moves, and it's affected by how well our condenser and whatever objective lens you're using can focus that light. So we can figure out the minimum limit of resolution or the resolving power. Remember, this is the minimum distance. Two objects would have to be apart from each other for you to see them as two separate and not blurred together. And so we can calculate this knowing the wavelength of light that we're utilizing, which of course, again, is a measurement in length, right, which is what gives us the distance component. And then the numerical apertures, which don't have units associated with them. And the condenser, which what focuses it onto your specimen. And then the objective lens, which is what's allowing you to see the image, right, which is taking that light in that's focused by the condenser, right, focusing that light so that you can see a clear image. Magnification. And again, we can calculate magnification because we're using compound microscopes. We're actually using two lenses, right? What two lenses magnify? The objective lens, depending on which one you're using. The ocular, the one you look through, right? So those times by each other give you the total magnification, right? So it's always the ocular times the objective lens. And that'll give you total magnification. And of course, this is a virtual image. This is not real, right? And it's increased so that we can help visualize what we're looking at. We're not actually increasing the size of the organism, right? Just the image of it that we're seeing. And then contrast is very important. So some microscopes can create their own contrast to help you differentiate the organism from its environment. Especially for bacteria, they're translucent for the most part, which means you wouldn't be able to distinguish them from the environment, right? It's like looking through a window, right? Sometimes they have to put those little stickies on glass door windows, right? So people don't walk into them, right? So you would pass right by a bacteria, you wouldn't even see it on your slide if we didn't create contrast. So bright field microscopy, which is what we employ in lab, right, doesn't generate any contrast. Unlike the name here, phase contrast generates contrast, or fluorescent microscopy, what's unique about the organisms, or what do we do when we're utilizing that type of microscopy? They fluoresce, right? So when UV light is shined on them, they fluoresce. So this is going to help in your visualization, or you use dyes that are fluorescent, right? So under those conditions, that would create the contrast that you would need. So we use stains, though, to provide contrast, because bright field microscopy is not going to provide any contrast. And the background is going to be quite bright, so you're not going to be able to distinguish. Dark field microscopy, little bit of contrast. Remember, the background is going to be dark and the specimens are going to be bright, but that's about it, right? You're not going to see any other level of detail there using that type of microscopy. So the stains that we use are going to help us see the actual shape and how they're arranged in three-dimensional space on our slides. So simple and negative staining you guys are going to do today. Negative staining is going to make it look like as if you're using dark field microscopy, because it's going to stain the background and not the cells. So the background is going to appear dark. Simple staining is going to stain the cells themselves with basic dyes. So you'll actually see the cells and the background will be bright, because we're using bright field microscopy. Gram stain and acid-fast stains are multi-stain step procedures that not only can you see the shape and arrangement of the cells, it's going to tell us something about the cell depending on what color they come out in this procedure. Right? It's one of two colors for these procedures because these are differential procedures. They differentiate one group from another. So for gram stain, those you guys in lecture should know the answer to this. What are the two colors? Purple and pink. Right? Because we use crystal violet and saffronin, which is a red dye, but will appear pink. right? So it's going to be one of those two colors, and that's going to tell us something about the composition of the cell wall for those organisms. Same thing with acid-fast staining. And then endospore staining and capsule staining, those are specifically for seeing those structures. Help us visualize an endospore. Help us visualize a capsule if they're present, right? Not all cells produce endospores or capsules. So, but if they are present, these special stains are going to help us visualize those internal for endospores or external structure for a capsule. So lots of staining, right? And as I said, differential, it'll help us differentiate things, right? What type of cell wall does it have? Does it or doesn't it have an endospore? So morphology, we have a lot of terminology related to the different shapes and arrangements, the groupings that will happen as they stay together after they divide. So the first picture is showing spherical cells. Remember, these are three-dimensional organisms, right? So again, for lecture class, this should be review for you guys, right? What's the term for this? Cox eye if you're talking about one, and if you're talking about all of them in that picture, cox us. I did it backwards. Cox us is singular, right? So this is Latin. US, right? Us, to us usually means plural, right? And I is singular, it's opposite here. So if you're talking about a single one, it's a coxus. If you're talking about more than one, it's cox eye, with the I. Okay, now this one, they're in pairs, right? They stay together in pairs. The term for this is diplo, meaning pairs, right, or two, and you would take into account that shape of the individual cell, which is coxus. So if you're talking about just one of the pairs here, it's diplo coxus. If you're talking about all the pairs, diplo cox I. Right? I don't think I put my I here. All right. This arrangement just refers to the four cells packed together like this in a square. You know what this is? This is tetrads, or tetrad or tetrads. Nice, easy plural that we're used to. Just add an S, right? Okay, this one kind of tricks people up. Okay, so we have part of the word, the term relates to the fact that they're forming a chain, right? Strepto, right? And they're individual cocci, so it's streptococcus or streptococci if you had more than one chain, right? The causative agent of strep throat is Streptococcus prurigens. When you say strep throat, you're basically just abbreviating the name of the bacteria that causes that infection. But it also helps, I find, to help us remember that strepto is a chain. Because I always think about when you have strep throat, how you would definitely not want to swallow a chain, right? It would hurt like hell. And, you know, we're talking about our throat that's inflamed, right? And it's kind of like a tube, right? So I think of that tube, I think of a chain going down that tube. That's how I keep strepto separated from the other prefix that it commonly gets confused with, which is the one for this cluster, which is staphylo. And then before that one, we have these packets of 8. This is Sarcinia. And then we have our staphylo, as I said, coccus. And as you can see, cocci can form lots of arrangements, right? We can have pairs, we can have groups of 4, we can have chains, we can have clusters, we can have these packet cubes of 8. Lots of choices for these cocci. Rods, not so much. The rods, most scientists just say rod because the term for this shape is bacillus, which there's also a genus Bacillus, Bacillus anthracis, right? That organism that causes anthrax is in that grouping of bacteria. So a lot of scientists just say rod shape, but the proper term would be bacillus, or bacilli if you're talking about more than one. Now, of course, here we have pairs, right? So what's our prefix for pairs? But these are rod shapes, so it would be diplobacillus or diplobacilli if we're talking about all the pairs. We got a chain, so now we're strepto, but the individual shape is bacillus, so this is streptobacillus. These guys are actually rods with a little bit of a curve. It's not the best curve, but you know, when you steal from the internet pictures, you can't be picky. What shape would this be? Vibrio. And they're showing a flagella on this one because a lot of the Vibrio, this shape, have a flagella. And so again, another genus name, Vibrio cholera, causative agent cholera, looks like this. This would be a long rod with multiple curves to it. So this is Spirilium, and a lot of them are motile biflagella, and this actually shows multiple flagella at either end. Spirilium for just one, Spirilia, instead of a UM and A at the end for more than one. And then this is all one organism, but it's not holding a particular shape. This phenomenon is referred to as pleomorphism, right? Or being pleomorphic, meaning that they have many shapes. And these cells are usually typically lacking a cell wall, and that's why they can't hold a shape. So Mycoplasma pneumoniae, a causative agent of a milder form of pneumonia known as walking pneumonia, is like this. It doesn't have a cell wall. It's pleomorphic. It doesn't hold a characteristic shape. These guys are helical, right? If you were to look at them three-dimensionally, under the microscope, they look like tight little squiggly lines, right? Where they'd be more curved to them for the Spirilium. So these are spirochetes, or spirochetes. It's pronounced either way. And they cause some pretty nasty diseases like syphilis and Lyme disease. So, as you can imagine, we don't mess with any of them, and it's hard to grow them and keep them happy in the lab. So guess what, y'all? The excitement stops with cocci and bacilli in this lab. That's it. That's all we work with. But, they can form different arrangements, right? We'll see some tetrads today, hopefully. We'll see some staphylo, some clustering, right? We'll see some individual rods. We might even see streptobacillus, some chains of rods. But, yeah, that's it. No Vibrio. No Spirilium, no pleomorphism, and definitely no spirochetes. So that makes it a little bit easier, right? For describing what you see under the microscope. So are bacteria all bad? No, bacteria can actually be quite good. The ones in your gut really contribute to providing vitamins for you and keeping you healthy. So there's tribes that they've studied in South America, right, that do things like roll around in the mud there. They have the most diverse microbial populations in their guts and associated with their skin. And some of the most healthiest people you'll meet in the world. They actually harbor bacteria that makes them immune to kidney stones, right? We've almost become too sterile in developing countries, right? We've limited our exposure so much to microorganisms that we've also limited our exposure to the good ones. Right? So then now what do you see on our shelves? What are they marketing all the time? Probiotics, right? They're microorganisms that have been proven to be beneficial, right? And we've limited our exposure so much to them that a lot of us don't have them, right? So we have to purposely take them to get these populations growing in our bodies. And then when we use antibiotics, do we kill just the bad bacteria? No, sometimes we kill off the good ones too. So you got to reestablish that microbial environment again after antibiotic treatment. So staphylococci, again, they're going to be clustered, right? We got some cool information on these slides, especially for you guys going into health profession, right? Where this guy gets his name is that it's actually commonly found on our skin, right? Epididymis, right? Referring to associated with our skin. Typically non-pathogenic if you don't have a compromised immune system. So some FYI information there. Then you have good old streptococci, which we know Streptococcus pyogen is a causative agent of strep throat. This guy is bad news. It can be major bad news, especially if it is a strain that produces toxins. So things like scarlet fever and toxic shock syndrome can be caused by this organism. Your immune system usually can clear up that type of infection, but usually none of us want to suffer through it, and so we go get antibiotics. And you really should get antibiotics because if you have a strain that has some of these other abilities, it can cause really serious complications. So not something to let go. The good news is something as simple as penicillin can knock out this gram-positive bacteria. Streptococcus pneumonia, another streptococci, really bad news one that can cause really serious pneumonia as well as other infections if it spreads throughout the body like meningitis. Vibrio, as I mentioned, Vibrio cholera, right, cause a really serious form of gastroenteritis, really bad diarrhea, bad situation. And then there's a type that can be found in shellfish, so it's one that they warn us about when you eat raw oysters, right, that you get an infection. But they do tests for this. They test the waters to make sure that the bacteria are not getting into the shellfish to begin with, and they randomly test the shellfish. But there's always the possibility, right, that you could get a bacterial infection from eating raw foods like oysters. So you're always warned against this. This is very rare, but again, even wading in waters that are contaminated, right, any wounds and stuff could also get infected, and that's what's shown here in this picture. So staining, why do we do this? So we can see them, so we can create contrast. What charge do our cells have? A negative charge. So most of the dyes we use are positively charged. They're attracted to that negative component, and they're going to stain the cells themselves. So what shape do we have here? It's a rod-shaped or bacillus, and then we even have some pairs there, and we've got some chains. You're always going to look for the most complicated arrangement that you see. And so in this case, the most complicated is a chain. So this would be streptobacillus, if you're trying to put a classification of arrangement on this particular picture. So some of those dyes that we employ in this lab, which are basic staining positively charged, are crystal violet, methylene blue, and safranin. What charge do basic dyes have? Positively charged. And so why are the cells stained instead of the background? Opposites attract, right? So negative cell, positive dye, they bind with each other. The other happens here. In the above picture, notice the background is stained. The cells are not. What shape? Can you guys see the shape of the individual cells? They're cocci, but again, the most complicated arrangement that we see over and over is the group of 4, the tetrads. So some acidic dyes that we use are Congo red, Eosin, which is also a reddish dye, and nigrosin, which is kind of a grayish dye, similar to what you see here. What charge do acidic dyes have? Negative. So why is the background stained? Yeah, like charges repel each other. If you try and take 2 magnets and you put them the wrong way, they just keep flying apart. That's because you have the same poles pointed towards each other. If you just flip it around, then they'll match up. But if you put the like charges, they literally just won't do it, right? They just keep flying apart. So in this case, the like charges repel each other. Negative for the acidic, negative for the cell. They just keep flying away from each other. So what makes a cell negatively charged? Anyone know what this molecule is? Nucleotides. The phosphates are what give it its negative charge. A nucleotide with a sugar, DNA or RNA, right? Do we have that in our cells? You betcha, right? And this is why, especially for eukaryotes, right, that nucleus stains really well, right? It's chocked full of DNA, negatively charged. It's very attracted to positively charged dyes. And then our membranes are made out of what?
Phosphate.
Yeah, again, phospholipids, right? So again, those phosphate groups that stick out, highly negatively charged. very attracted to the positive dyes that we use. Where the fatty acids, on the other hand, these don't have any charge. But those are inside the layers, right? Little note on trans fats, right? And why they're bad. So what's the consequences of leaving a stain on? And we're talking about simple staining using a basic dye, right? So remember, this is going to go where? Into that cell. It's heading towards that DNA. It's heading to the membrane to... All staining kills. They'll burst. They'll burst, right? They'll burst. Or they'll really swell up so much so that, especially if you have cocci and they're in a group, it's hard to tell. Are you looking at tetrads? Are you looking at... What are you looking at, right? Because they'll kind of blur into each other, right? So the bad news with that is that their morphology is going to be distorted. They're going to appear larger than they should, right? And it's going to be hard to distinguish cell from cell if they start blurring into each other. And they may even burst, and then you're not going to see anything. What if you don't leave the stain on long enough? Yeah, you won't be able to see them. The whole point of staining them, right, is to be able to visualize them. If you don't stain them enough, you're not going to be able to easily visualize them under the microscope. I always say in microbiology, when in doubt, stick with 1 minute, right? A 1 minute rule pretty much works for staining. For most stains, if you do it for a minute, it's good enough. So when we stain them, we could look and see, as I said, rods and cocci. And based on their shapes, really affects how they interact with the environment. So if you were to have a rod and a cocci that had the same volume, right, same amount of stuff, because of this structural difference of being shaped as a sphere, right, as opposed to a rod, they have a different surface to volume ratio. Meaning that when you look at the calculations of it, a rod has 4 as its ratio, where a coccyx has 3. So a rod has more surface area compared to its volume, right? Because it's at a 4, where the cocci is at a 3, where they have both the same volume. So the advantage here is what happens at the surface of a cell? Things come in and out, right? If you don't have a lot of surface area, it's hard for stuff to come in and out, right? Thank you for whoever did this commercial. Is it Cox, right? They get all those people, right, and a little tiny hole, right? It's just like the internet, right? Everybody wants to get through that hole. Well, if you only have so many holes, only so much stuff is going to get in and out, right? Unless you open up the hole, right, and everybody can go running through. So if you've got a small surface area, you basically have a small door, right? And everything's going to fit through that door. That can get kind of clogged up sometimes. But if you have a large surface area, you got a big door, right? No problem getting stuff in and out. Make sense? And that's very important for a cell. So, you know, a rod on average has about 160 times better, right, travel of stuff in and out. So, you know, we can mathematically calculate these things, and Peter loves to get into the math, right? I don't, right? So all the math is here, right? For those of you guys that love mathematical formulas and all that jazz, but I'm not going over it. But basically the best we can do is a cylinder when we're talking about rods. No testing on formulas for this, right? There's one special case where the volume of a cylinder and a sphere, right, are the same. And again, that's where we can compare that surface to volume ratio. So the diameter, right, So how thick that sausage is, can you imagine it that way, versus its length? This is the approximate upper limit, right? They really don't get any bigger than this, right? Although this would be great, really not the case. So a disc, this would be more like a disc where it's wider, right, than it is high. So our red blood cells are like that, right? They're discs, are they not? They're actually biconcaved, right? Greater surface area for the gas exchange that needs to happen. So now we're going to play with Play-Doh that I stole from my son. He doesn't need it. I just made a mess with this stuff. So get a little bit. Oh yeah, you don't need your gloves until we get going. So divide what's in the container up. Everybody get a little piece of Play-Doh. So I want you to take your piece of Play-Doh, and then whatever piece of Play-Doh you have, I want you to divide it equally in half. This is going to be our same volume, right? One of them make it into a ball. This is going to be our what? Our coccyx, our sphere, right? And then the other one make into your rod, your bacillus, your little sausage, right? Bet you didn't think you were going to do this today. Back to our preschool days, right? Kindergarten. But because, remember, these organisms are three-dimensional, right? I can't do that up on the TV screen. So, same volume, right? Lots more surface area compared to that volume of this rock. And then I could have fun. Ooh, it's getting dried out, huh? I'm gonna have to invest in some. Huh? I actually already bought some for his project and we didn't end up using it, so I actually have some stashed at home I need to bring in. Alright. What's that? Vibrio. What's that? Spiralium.
This is harder. You're trying to do the helix shaped DNA spring.
Alright, we're done playing. Put it away.
I know.
I just make a whole bunch of fun shapes, right? Alright, some more shapes. Which one's going to cook faster and why? Which hamburger? The thin one, right? Why? Greater surface area to volume. Which sausage? The thinner one, right? Greater surface area to volume. So heat distributes faster. It cooks more quickly, right? You're going to have to really wait a while for that cute little meatball and that thick hamburger, right? Slow heat transfer. Some of us were doing that over the weekend. So again, a lot of numbers, but what's the take-home message? Right? A sphere, no matter its size, its surface to volume ratio is going to be limited. And so this is why pretty much the spheres are so tiny, right? Because the bigger they get, you're going to have more volume and not enough surface area to support it, right? Just not enough to be able to be happy. So, graft, right? You're stuck on this one little line if you're a sphere, right? Doesn't get any better than this. Where, variations based on how long, right, and thin of a rod you are, the better off you're going to be. So, organism level. How do these guys achieve this whole surface to volume problem? Yeah, they're branched out. And the leaves themselves, right, are flat. Right? Greater surface area to the volume that they're supporting. Right? These cute little needles, same thing. Great surface area. Even better insulated. They're nature's solar panels. Right? That whole concept of you got to have enough surface area to get the job done. In this case, photosynthesis and gas exchange. Right? But the same issue, right? You got to have the surface to get it done. To support that volume within. So, consider a coccyx and a rod of equal volume, right, that we just did. Who's got the better surface area? The rod. But we're in a dry environment. What are you going to lose in a dry environment? Moisture. Can you afford to lose moisture? No. So in this case of this type of environment, who is better suited? The sphere in this case, the cocci. Because it has a smaller surface area, so it's going to lose less water. So cocci actually do better in drier environments, right? Because of that less surface area. So environment, right? So when we're looking at it, you're like, oh, every bacteria should be a rod, right? More surface area. Well, there are conditions where having a big surface area is not so good, right? When it's dry, it's better to be a cocci, not lose so much water. Right. The more exchange, but you're dealing with the problem that because you have a higher surface area, you're going to lose more water. Right? So the cocci is better suited because he's not going to lose as much water. He's still struggling with the issue of not as much surface area. But there's enough to get the job done, otherwise cocci wouldn't exist, right? But there's a reason evolutionary-wise why they probably still exist. They are better suited to dry environments than the bacillus is. Now, if the environment's moist, of course, who's going to outperform the bacillus in this case? Because, again, of the greater surface area, more exchange can grow faster. And you don't have to worry about water loss. But remember, rods can be stretched out into vibrio, curved rods, or spirillum, or spirochetes. Right? That's all just a rod. It just has, bumps. As I demonstrated with my Play-Doh. So the same thing, right? They have a very large surface area based on a small volume. So negative staining. So this is where we're going to use acidic dyes. So why doesn't a negative stain colorize the cell? Right, the opposites repel. We're going to use negative staining employs acidic dyes, which are negatively charged. Cells are negatively charged. They repel each other. Only the background gets stained. So, Eosin, which I think they didn't tell you, so I added from the questions in your book, is an acidic dye. So what charge? Negative. So what's it going to stain? Background. Methylene blue is a basic dye, so what charge? What's it going to stain? It's positively charged. What's it going to stain? The cell. So what are you going to end up with? A blue cell with a red background. We're going to employ this type of utilization of charges for staining backgrounds in cells when you guys do your capsule stain next week. One of the dyes we use is an acidic dye. It'll stain what? It's an acidic dye. The background. We're going to use it today. It's called Congo red. The background, the most of your slide is going to be red. But then we're going to go one step further because we want to stain the cell as well. right? So we're going to use a reagent that's going to stain the cell, and in this case it will also stain the background. I have never played around with this, what they've suggested here. I think it would matter what order you did this in as to, you know, whether you would then again stain, say, the background or the cell. Yeah. Yeah. So, but what we're trying to get out of you with this question is remembering the fact that basic dyes are positively charged. They're going to stain cells. Acidic dyes are negatively charged. They're going to stain the background. Right? And sometimes this is going to be beneficial. Sometimes we want the background stain. Sometimes we want the cell stained. Or in the case of the capsule stain, we need to stain both to see the capsule that will not stain because the capsule is not charged. Right? We can't get any dye to stick to it. It doesn't have a charge. Make sense? All right. So it's all about the charge. So if you were to compare Micrococcus luteus stained using the basic stain procedure and staining it using an acidic stain, a negative stain procedure, what might account for the differences? So what will actually happen is your basic stained ones are going to appear smaller than your negative stained. And you're like, huh? Why would that be? What's a procedural difference that's different between these two procedures? What do you do in one of these and not the other? Heat fix. Heat fix. When we prepare a slide for basic simple staining, you must heat fix it first. You must prepare that slide for the staining. In that preparation, you heat fix, which means you heat them so that they will stick to the slide, right? So proteins, when you heat them, become tacky and sticky, right? This is why before non-stick frying pans, thank you, Jesus, right? You always had to put some type of lubricant before you went to go fry your egg. Otherwise, it would stick, okay? That's because when you heat up those proteins, they become sticky, and they would stick to your pan, okay? So what are we going to do to these cells? We're going to make them sticky. So they'll do what? Stick to the slide. And that way when we do rinse, they don't wash off the slide. When we negative stain, we don't heat fix. Because we're going to put the cells right into the dye and spread it out and just let it air dry. For basic staining, we're going to flood them with stain and rinse. So, but the major difference that's going to affect size here is the heat fixing. So what do you notice about that egg when you fry it? What happens to the size? It shrinks. That's why the basic stain ones appear smaller. They get shrunk by the heat. What does the heat also do? It'll kill them. That's why we heat our food. It's kill microorganisms. That's why you heat it to a particular temperature, right? To kill particular organisms that may be present in your chicken or beef or fish, pork, right? So we heat to make them stick. So you ready to make some sticky slides? I am.